All right, so today we are going to be talking about cells, and this is A2.2 in your textbooks, and we're going to be looking at what are some common features of cells and some oddities. So when we're looking through the slides for bio this year and next, you'll notice there's a bit of a heading here, and then often there are page numbers either here or in the top right corner of other slides. So the theme of this unit is unity and diversity. And so what we want to be thinking about here is how are cells the same and how are they different? So cell theory, first off, is going to tell us that all living things are made out of cells. Cells are made from pre-existing cells, and cells are the basic unit of life. Common features for all cells is that they have genetic material and proteins. They have membranes which control entry and exit of substances into the cell. They produce energy, and their activities are catalyzed by enzymes, which are also proteins. So all of this is happening in the cytoplasm, or a lot of it is happening in the cytoplasm, which is where metabolism is going to happen. And metabolism is this enzyme catalyzed reactions. But of course, our genetic material is going to be in here in this purple circle, which is called the nucleus. So some cells are a little bit abnormal. It's not that they don't follow cell theory, it's just that they don't have all of those common features that we were talking about in the last slide. So one example here is skeletal muscle tissue, which is right here. They're much larger than normal cells, and they have many nuclei in one cell. Typically, a cell will only have one nucleus. Phloem sieve tubes, that's these pink ones here, and they have a nucleus and different organelles initially, but those break down, leaving only the outside membranes here, which are the outside edges of these pink tubes. And then they're connected to helper cells, which are called companion cells. And those helper cells do have organelles and nucleuses and can do different jobs. Because there's nothing in these tubes aside from membranes, they can't really do very much other than transport things that other things have put in them. Aseptate fungal hyphae, which are these blue ones up here, are also quite large. And if it's septate, a fungal hyphae, they have these divisions and they end up having one nucleus per cell, but in aseptate fungal hyphae, they don't have the divisions, and then they end up with many nuclei in one cell. And then red blood cells, which have no nuclei. So that allows them more space to carry oxygen. So there's two types of cells that we need to know about, and those are prokaryotes and eukaryotes. And prokaryotes are structurally quite a bit different than eukaryotes. Prokaryotes are bacteria, and eukaryotes are the cells that make up, for example, humans or plants or animals, or even amoebas, which are eukaryotes but are single-celled. So prokaryotes are small, much smaller than eukaryotes, and they're bacteria. They have a simple cell structure with no compartments and no membrane. So inside the bacteria, there's no membrane structures. But on the outside edges, there are. So there's obviously a membrane on the outside, because otherwise everything would be flowing in and flowing out of the cell, and it wouldn't be able to maintain its integrity on the inside. DNA here is circular, but it's not in a nucleus because it doesn't have those inner cell membranes. And so this blob of DNA is called a nucleoid. DNA does not have proteins to help it coil. Usually for eukaryotic cells, they're going to have proteins that are going to help the DNA to coil, and that allows it to separate properly when the cells are reproducing. Prokaryotes have a cell wall that helps to maintain structural integrity also, and then they contain these little segments of DNA called plasmids that are separate from the nucleoid region. And plasmids are they actually give prokaryotes their superpowers. So plasmids are where you would find genes that are responsible for resistance to antibiotics or resistance to different viruses. And so plasmids actually can be swapped. Bacteria can share them, or they can pick them up, scavenge them from dead bacteria. So that's how drug resistance to antibiotics happens. The proteins my goodness, the ribosomes inside a prokaryote are smaller than inside a eukaryote. They function the same, but they're just structurally a bit smaller. So we call those 70S ribosomes. And then prokaryotes reproduce by binary fission, which is a way of cloning themselves. And this process is kind of like creating a little mini me. Once they're big enough, they just split in half, and that's it. So then structurally, what's the difference for eukaryotes? Eukaryotes have DNA and a nucleus. Their nucleus DNA is not naked. So the nakedness refers to proteins that are called histones that help that DNA to coil. DNA is linear, not circular. It was circular in the nucleoid region of the prokaryote. And we know this because when we see pictures of chromosomes, they're all little straight lines. So there's 23 pairs of them in humans. That number differs across species, but what that means is those little lines of chromosomes that's the DNA material. So there's 23 separate strands of them. All the organelles inside a nucleus are separated from the cytoplasm by membranes. So you can see all of these little membranes on the outsides of things here. And that allows each compartment to do different jobs. And if it is a plant cell, it will have a cell wall, but not all eukaryotes have cell walls. So animal cells do not, for example. They have 80S ribosomes. Ribosomes are responsible for making proteins, and these reproduce by mitosis. So that's a way of replicating the DNA and then splitting. So it is cloning themselves, but they use special structures that are not present in prokaryotes when they reproduce by binary fission. So processes of life, we can remember this by Mr. H. Grin and the acronym for it. And so we have two organisms here. These are both single cellular eukaryotic organisms, but even at a single celled level, the paramecium here and the cladomonas are both going to do these different things. So they're going to have maintain homeostasis, so they're maintaining constant internal environments. In the case of the paramecium, it might do that using this contractile vacuole. So it's going to squeeze out water when there's too much water inside the paramecium. And it can also squeeze out water through the contractile vacuole to make it like shoot forward. They both have metabolism, so those are biochemical reactions happening in their little cell bodies. They are going to take in food, so nutrition, and that food is going to supply nutrients for growth and repair, and that food is going to be broken down into energy that is able to be used for this growth and repair. They're going to create metabolic waste. In the case of generating energy, that's going to be carbon dioxide, but in the case of breaking down proteins, that might be urea. That's the case in humans. That's what metabolic waste product, and we have to remove that. And excretion would be breathing out, or it would be peeing for humans. Obviously, parameciums do not have bladders, so they're not peeing the same way that humans would. They're going to grow, which is an increase in cell numbers. So they're going to replicate themselves, and they're going to respond to stimuli. So they're going to perceive their environment, and then they're going to swim around in it as a byproduct of that. So they are going to be able to perceive threats, for example. And then lastly, they're going to be able to reproduce. So they're going to create offspring, and that might be done sexually or asexually, meaning they have a sexual partner that swaps DNA with them, or they just clone themselves, but they're able to reproduce. And that's going to be different than growing. Growing is just going to be making one cell bigger, but reproducing is going to change one cell to two cells. All right, so eukaryotic structures. Some of them here that we're going to see, I don't know if I can move my head here so that it's out of the way, but so plastids, these are only in plants, not animals. These are, you've probably heard of them, more commonly known as chloroplasts. So they are what does photosynthesis. Plant cells will also have cell walls. Plant cells will have cell walls. And so these eukaryotic structures are structures that can exist in eukaryotic cells, but they're not necessarily going to exist in all eukaryotic cells. Vacuoles, which are small for animals, but they have one centralized large one, which is here for plants and fungi that stores water for the plant because the plant can't just walk over and get some water from a lake. Centrioles are these protein tubular structures. These are only in animals, and they're used for cell replication through mitosis. They're not for plants or fungi. And undilopodia, so that would be cilia or flagella. So these little hairs here are cilia, and then flagella is this tail. These are occurring in animals, but not plants or fungi, except for, in some cases, the reproductive cells, which are called gametes of plants or fungi, will have some undilopodia. These tail or hair-like structures allow movement of these cells. And then lastly, what we're going to talk about is cell size. So as cells get bigger and bigger and bigger, you'll notice here, well, maybe you won't even notice, but what ends up happening is the surface area to volume ratio of these cells gets smaller. So cells can usually only get so large before they're unable to function effectively. They're unable to get enough nutrients in and get enough waste out. And then you end up with not enough energy and too much goop inside the cell, and then the cell is going to become contaminated and die. So this is because the surface area to volume ratio changes as the size of the cell increases. So when we are looking at this first 1 by 1 by 1 cube here, its volume is 1 centimeter cubed, and its surface area is going to be 6 centimeters squared. And that's because we have 1 by 1. So that's 1 centimeter squared, but then we have 6 sides. So we add those up together, and that's going to be 6 centimeters cubed. And our surface area to volume ratio is going to be 6 to 1. But when we look at this 2 by 2 by 2 cube, multiplying these three numbers together, we get 8 centimeters cubed for our volume, but then we have 24 centimeters squared for our surface area. And when we have a ratio of those things, it's going to be 3 to 1. So this is a much smaller surface area to volume ratio. And then that continues to happen with the 3 by 3 by 3 cube, where our volume is 27 centimeters cubed, but our surface area is 54 centimeters squared. And so it becomes a 2 to 1 surface area to volume ratio. And so the volume is getting much bigger more quickly than the surface area is, because volume cubes and surface areas use squares. And as a byproduct, not enough stuff can come in and out of this cell in order to accommodate for all of the things going on in the cell. So cells that have a high surface area to volume ratio to allow them to function properly are going to be red blood cells, proximal convoluted tubules, these reabsorb things in the kidneys, and then alveoli in the lungs to facilitate gas exchange. And here you can see on this elephant, I included it here because it has a very large surface area to volume ratio on its ears, and that allows heat to dissipate from its ears to allow the elephant to cool itself. So to sum up, cells usually follow cells always follow cell theory, but there are some exceptions with regards to their structures that are common, but not always there. And we looked at red blood cells, sieve tube cells, and skeletal muscle cells as examples of that. Eukaryotic cells have much more complex DNA in a nucleus, different sized ribosomes, and compartmentalized organelles that carry out specific function versus prokaryotes. And then there's processes that are carried out by the cells, and those include metabolism, homeostasis, nutrition, excretion, growth, response to stimuli, and reproduction. And that is all for today. Hopefully I can